Hi, everybody, and very glad to be talking to you again. Very welcome to the 21st episode of our discussions and our broadcast, the Brain Function series from the Togetherness Media, Los Angeles. Mohammad Nami here. I'm a medical doctor, cognitive medical neuroscientist, and sleep disorders fellow. And like ever, I'm so privileged for the opportunity of talking to you regarding the significance of our brain and cognitive function. And as you probably recall from the earlier episodes, together we kind of, guided the discussions up to a point that we arrived at a significant keyword, which was neuroplasticity. And we kind of agreed that this keyword is very fundamental in a way that in many different realms and perspectives of neuroscience and studies of the brain and cognition, we come across this idea of neuroplasticity. And that means how the brain reforms, reorganizes its structure, and gains new functionality, gains new patterns of strengths and functions. And these all relate to the pattern of connectivity and how neurons are extending new projections, how neurons are forming new synapses, and how those synapses are supposed to be stable, how they are interacting in a sense that a new network. But before a network, a new circuit, and then based on the cumulative function of the circuits, new networks will form. And we kind of agreed that when networks are being employed simultaneously, when we're involved in a complex function or assignment or task or something we're doing, then we might be having a concept which is called meta-networks, which means the network of networks. So how these different patterns and areas and also, connectivity meshworks in the brain, they come together and they are just running a quite orchestrated series of functions where we'd be able to remember, to learn, to pay attention to something, to talk, to regulate our emotion, to go for decision making or reasoning or planning, and a range of executive functions and so on and so forth. So that's pretty amazing, right? So the studies over the concept of neuroplasticity for the past 20 years has dramatically been increasing. And we've been a booming number of papers in the scientific literature that they are kind of highlighting the mechanistic underpinning pathways beyond neuroplastic changes in the brain, in a sense that today we acknowledge the fact that neuroplasticity is not only happening in the brain, but wherever that we have the neurons, there is a chance for plastic changes in the neural connectivity, in the neural dynamics. So even in the peripheral nervous system, we know that we got degrees of neuroplastic changes. So the more we use a specific like extremity, or we're involved in a certain task, by that area or that function would not only get enhanced in the brain, but also in the peripheral nervous system. But something is really important here, that we have different dimensions of hypothetical views and also explanations and justifications to explain the process of neuroplasticity, in as much as many scientists are not totally agreed with these kind of things. So there's a lack of concurrence and agreement between scientists with regards to the different explanations and also theories that we already have for the idea and the phenomenon of neuroplasticity. But the mostly acknowledged mechanism and idea and theory behind neuroplasticity is neurobiological theory, which is mostly agreed by many of the scientists. So first, two scientists, like back in 2002, 2003, two scientists, which were T.J. Wall and also Dr. Zhu. They were the first scientists to put the idea forward that, well, we can explain the neuroplastic changes in the brain from the neurobiological standpoint. So they discussed about what happens in the neural projections in the in those specific projections, when we have the extension of this podocytes or specific projections in the neuron, when two neurons are about to extend new projections and form new synapses, then firstly in those neural projections we have some reformation and restructuring of the cytoskeleton. And as we know, the cytoskeleton is formed by the microfilaments and neurofilaments, and they are made of tubulins. And tubulins are proteins. These are peptides. So we have tubulins and many other peptides that they are forming the cytoskeleton of the neuron. And what causes the production of those peptides is the expression of the gene, and that sits within the nucleus on the genetic material of the cells of the neurons. So everything just relates back to the genetic changes and the expression of certain genes. So by that, when some new behavior in the cell is formed, as just established, some new synapses are there, and some new projections, some structural changes, some dynamic alterations within the body of the cell, within the genetic materials, and specific genes are being expressed. So all the way back to the gene and forward to what we see in the specific synaptic changes, what we call it synaptic plasticity. We know that the synaptic plasticity and the neuroplasticity does not happen only on the cortical level of the brain, but when we look at the different structures, hierarchically positioned within the brain, we have the white matter and the tracts and fascicles, and how the neural structures in different parts of the brain are getting connected. Also, we have all the way up this hierarchy of functions. We reach up to the neural layers at the cerebral cortex. So we know that the neuroplasticity might happen across these different structures and layers of functionality and organization. And we got different types of plasticity. One type of plasticity is literally like structural. And this is when we do the CT scan, when we do the MRI, we see some changes in certain parts of the brain, that the thickness of that structure, the thickness of that part of the cortex is getting increased, or we know that the volume of a certain nucleus in the brain, for example, amygdala or striatum or caudate, for instance, the volume of that certain area, of that certain subcortical region, is increased or decreased. For instance, when we're talking about the neurodegenerative changes for Parkinson's disease, or when we're discussing dementia of Alzheimer's type, we see that the height of the hippocampus is decreased. So the volume of the hippocampus, the size of the hippocampus is changed, and that has been shrunk. All right. So by that, we know that there has been some detrimental changes. There have been some cell loss in that area. So by this, we have the shrinkage of the structure. So the volume is negatively, affected. And this is structural change, okay? And on the other hand, we might be having some new cells and new formations. And that might not only happen during the childhood or early adolescence, but we have the process of neuroplasticity and the formation of the neural stem cells, perhaps across the lifespan. Earlier, we only believed that there are some neural stem cells being generated or proliferated in specific areas of the brain, including the dentate gyrus within the hippocampus and also in the olfactory bulb. But nowadays, we believe that some other areas, including the cerebellar hemispheres, they also have the capacity of generating and proliferating some neural stem cells at certain levels. And we know that by those things, they might get the capacity of repairing themselves or their adjacent areas, or even the distant areas of the brain might at some level receive some inputs from these areas that they have the capacity of generating neural stem cells, all right? And so the thickness might decrease, might increase, and also the volume may decrease, might increase, and the neural wiring is getting changed. And that is exactly what we are referring to as the structural neuroplastic changes. But is the neuroplastic change only structural? No, that's not the case. We also have the functional neuroplasticity. And the more we use a certain part of our brain, the more we use, the more we employ a function or like an interaction with the environment as being biased toward a specific task, that area in the brain is getting even strengthened and more strengthened, more strong by time, and it will get strengthened. So the thickness, also the structure, the dynamics, and also the stability of the synapses will get positively, affected. So this is what we call it LTP, or long-term potentiation. But on the other hand, we have LTD, or long-term depression, when we do not use a specific function or a particular, action or skill. Then by time we're going to lose that kind of, functional capacity. This means that the synapses will be negatively hampered, and we have the long-term depression. Sometimes we use we repeatedly recall some memories or some learned motives. So by that, the synapses that we're using more will stay longer and more stable. But on the contrary, when we're not using some certain task, we're going to lose those tasks or capacity of performing those tasks, or lose those synapses or circuitries. So we have the option to either to use it or to lose it. And the more we use our brain, the more brain will certainly get to use. And that is called the cognitive reserve, or the functional reserve of the brain. And many functionalities, domains in the brain, including the memory and learning, and also some emotional, sensory, motor, and behavioral functions in the brain, are subjected to the concept of neuroplastic changes. So by time, we might get improved by a specific predicament or impairment or injury that we already suffered from. And also we might enhance or empower a particular part of our functional capacity at a certain level. And so we need to know more about the dynamics in the brain and how neuroplasticity alters this dynamic, and how this is going to change the process when we're recovering from a disease or injury or predicament, when we have issues with, for example, blindness or deafness, or people who have gotten neurodevelopmental delay, or like issues with mental illnesses. This is all relating in some ways that is related to neuroplasticity. So we got to better understand the dynamics of neuroplasticity and the function and the benefits that we need to obtain new skills through which we would benefit the brain. We would positively affect the brain function. All right. So this is what we were supposed to talk about it today, and in a nutshell, what we discuss is the homeostatic changes in the brain as per the need when we're using a certain task, a certain skill is being getting stronger by time, and we got to be talking more about the implications and also the way that we're applying neuroscience insight and also neuroplasticity science into human empowerment and also improvement of the deficiencies or impairments in the function in the brain. So by next week or the following sessions, we'll hopefully get a chance of discussing around the implications of neuroplasticity, and this is what we're supposed to tap into. And again, I'd really like to thank you again for turning in and for your kind attention, your patience listening to me. And I hope that this is the way that we're going to find new horizons in front of us, the new insights, new questions, and new opportunities for shared interests and explorations of the miraculous organ, the brain. Until next week, I hope that you stay well and take good care of your brain, and we'll catch up later. Thank you.
Togetherness Media. Togethernessmedia.com